When I think of epidemics spreading through a human population, especially a new virus that's never been in the human population before, the image in my mind is like a forest fire spreading through an old forest in the West. It's old and it's dry. There's just a lot of combustible material there. And that's the same thing that happens for a new virus entering the human population. When a new virus enters the human population, none of us have immunity, so it can really spread like wildfire. But as it's spreading, what happens is it's basically running out of fuel. It's burning through its supply of susceptible hosts as hosts become immune to that virus, and so then the virus has to do something. And what a lot of viruses do is they mutate. By mutating and escaping immunity in the host population, they're essentially adding fuel to their own fire. I never really expected that I would become a virologist. I was actually really interested in plants when I was an undergraduate studying biology in college. But when I first got to graduate school and met my future Ph.D. advisor, Dr. Katja Cole, I became completely obsessed with viruses. And that's partially because Katja is a very charismatic, brilliant scientist, and also because she was studying influenza in a way that completely blew my mind at the time. She was trying to model influenza in a way that considered both the evolution of the virus, as well as how the virus's epidemic dynamics, how many people get infected with the flu every year, depends on how the virus evolves. There's actually this really intimate, neat feedback between viral evolution and how many people get infected with influenza each flu season. And this idea that flu was evolving year by year, really on timescales of weeks or months, really got me interested in viral evolution, because we normally think about evolution occurring over millennia or several eons. And here we have this virus that's evolving every year to escape immunity in the human population. And that got me really excited about influenza and studying virology in general. Of course, at the time, I was, a brand new graduate student, and I knew really nothing about epidemiology or how to model infectious diseases. So I was really nervous about asking Dr. Cole if I could join her lab, but fortunately she said yes, and I've never regretted joining her lab because it turned out to be a really great experience. And I think it's really important for you all to keep that perspective in mind. If you're taking a class here at NC State and you meet a professor who you think is doing really neat, interesting research, you should, by all means, ask that professor if there's an opportunity for you to participate in that research. Oftentimes, professors will say yes. And I have to say, as a professor myself, it really never gets old having students come up to you and say to you that they find their research exciting or interesting. So take a chance. If someone's research really sparks your interest, ask them if there's an opportunity for you to get involved. So a really hot topic in public health and infectious disease epidemiology right now is what we refer to as genomic epidemiology, or sometimes genomic surveillance. And what this means is that we go around the world and we collect strains of different viruses or different pathogens from different infected individuals, and we sequence those pathogens or those viruses so we can see particularly what strains those individuals are infected with. One of the things that really got me excited about influenza in particular at the beginning of graduate school was just how much new genomic data there was becoming available for this virus in particular. Around that time, it was amazing if we had tens or even dozens of sequences for any particular virus. But in the case of influenza, hundreds, and probably now more like thousands of influenza strains are having their whole genomes be sequenced every single year. And that gives us a lot of information about how the virus is evolving and how the virus is moving around the world. So this type of genomic surveillance knowing exactly what strains are circulating in the human population turns out to be incredibly important in its own right. Knowing exactly what strains are out there gives us an idea if there's a particularly virulent strain circulating somewhere in the human population. So when we talk about the virulence of a particular virus, what we mean is how aggressive that virus is in terms of infecting the host cells and causing damage in the host body. From these influenza genomes, we can also study the evolutionary dynamics of the virus. We can reconstruct viral phylogenies that show how all these viral strains are related to one another. So what is a viral phylogeny? Well, just like any other family tree that shows how different members of a family are related to one another by descending from common ancestors, viral phylogenies show how different viruses are related to one another through shared lines of descent or through this genealogy. And if we can reconstruct these viral phylogenies, we can also learn about how the virus is spreading through the human population. We can think about every single lineage or branch in the phylogeny as telling us a little story about how the virus is spreading from location to location. So now if we have these viral phylogenies that contain hundreds or thousands of different strains, what we have is essentially hundreds or thousands of different stories that show us different paths that the virus has used to spread all around the entire world. It also gives us an idea if the vaccines we have against the influenza virus are going to be useful and actually protect us from getting infected, because we can often see ahead of times if our vaccines are not particularly well targeted against particular influenza strains in the human population. Viral phylogenies contain a tremendous amount of information about hundreds or even thousands of different viral lineages and how those viral lineages have moved from place to place throughout the world. Air traffic flow, that is how many passengers get on planes traveling from city to city, is actually a remarkably good predictor of how a new virus will spread around the entire world. So for example, in pre-COVID-19 times, if we knew that a new virus, say, emerged in Singapore, we could use air traffic networks and how many flights and how many passengers each day moved from Singapore to any other city in the world to figure out how that virus would spread from Singapore through the rest of the world. Of course, if this virus was particularly virulent, like COVID-19 was, people would very quickly figure out that there was a new virus spreading throughout the world, and so our entire air traffic network would change. There'd be less passengers flying between cities, and flights would eventually get canceled, as we saw with COVID-19. And so this makes long-term predictions difficult. It's hard to predict how humans will adapt or respond to the presence of a new pathogen. But what we can get from these viral phylogenies is insights about how viruses will move through the human populations when they first arise. So the big question is if we can use viral phylogenies to predict how a virus will spread throughout the world, can we also use viral phylogenies to say something about how the viruses themselves will evolve in the future? And I think we can if we're realistic about the timescales of our predictions, just how far into the future we think we can predict. There's been a lot of recent interest in what we call short or near-term predictions, because actually a lot of important public health decisions can be based on these short or near-term predictions. For example, we might be interested in whether antibiotic resistance to a particular drug will evolve in a pathogen population in the near term or not. But I think influenza provides another great example of how we can predict the near-term evolution of a virus. So many of you have probably had the unfortunate experience of being infected multiple times with influenza virus. And the reason why humans can get infected with the virus again and again is because the virus can actually escape our immunity towards that virus. So when we get infected with a particular viral strain, we develop strong immunity to that particular strain by developing antibodies that can target and bind to that particular viral strain and prevent it from entering our cells. But what happens, and what we've seen a lot already, is that viruses are constantly mutating, constantly evolving. And some of these mutations will change the virus's proteins that human antibodies bind to and recognize. And when these new mutations occur that can escape our immune systems, those viruses are going to have a huge advantage because they're going to be able to infect a bunch of new hosts that previously had immunity to this strain. And so those strains carrying these escape mutations are going to then spread all throughout the world and then potentially infect a bunch of new hosts. So how good are our predictions? Well, admittedly, they're not super great. We only pick the right strains about 70 to 80% of the time, which is why sometimes we still get suboptimal vaccine strains. But I think we're getting better at these types of predictions all the time. At the end of the day, making predictions and the whole business of forecasting the future is really about information. If we don't think our predictions are very good, we need to figure out what additional information we need to make better predictions. I think in a lot of ways, our current predictions about flu are kind of like early weather forecasts. Even in the 19th century, scientists already understood a lot about the physics driving the weather, about how atmospheric temperatures and pressures change the direction of the wind and so on. But the infrastructure to gather and process the information wasn't there, so it took another 100 or so years to even get a reasonably good 5-day forecast. However, once we had satellites and other technology to measure atmospheric conditions throughout the world, and the raw computational power required to process all that data, accurate weather forecasting became possible. And as soon as we were able to make these predictions, we could begin to refine them through an iterative process of checking our predictions for the next day's weather against what we predicted the day before. I think the same thing is now happening with our flu evolutionary forecast. We can check our predictions from the previous year against what we see in the next flu season, and if we guessed wrong, we can update our models to try and make better predictions. And since we can do this every year, iteratively checking our predictions and updating our model, I think our predictions will improve quite rapidly. I think the key here is to think about what information we need to make better predictions about how flu will evolve. For predicting viral evolution, we at a minimum need to know what strains or genotypes are out there and what mutations these strains carry. And we need to know the fitness of these strains if we're going to predict anything about their fate in the human population. So we really need information about the fitness of each strain, how competitive and how transmissible each strain is relative to other viruses. But measuring fitness is notoriously difficult. For example, we can measure the fitness of a particular virus in the lab and see how well that virus replicates in cells in a petri dish. But that tells us nothing about how the virus will actually spread throughout the human population, because a virus can be very good at replicating in cells at a petri dish, but might completely fail to spread through the human population. So we really need to know about viral fitness at the population level, how well the virus can transmit and infect large numbers of new hosts. Some of the best predictions for flu virus have actually used viral phylogeny as an indicator of how fit different influenza lineages are in the human population. More fit viruses will be transmitted more often and infect more people. We can see this in the viral phylogeny itself because more fit viral lineages will branch more often and have more sampled offspring or children, which you can see at the tips or the leaves of the tree. So for influenza, scientists have actually been able to get pretty good estimates of how fit different flu strains are in one flu season by looking at the rate at which different influenza lineages are branching in the phylogeny, and then using these fitness estimates to predict which strain will become dominant in the next flu season. The thing that makes predicting longer-term viral evolution so difficult, beyond just the fate of strains that are already in the human population, is that we don't know what mutations will occur, and so what the different strains will be in the future. So in that case, it's like trying to make a prediction without even knowing who the major players will be. What we do know is that SARS-CoV-2 is certainly evolving in the sense that new mutations are constantly occurring and creating new strains or genotypes of the virus. That can make viral evolution sound really scary, but the vast majority of these mutations are neutral, meaning that they don't affect the virus's fitness, or may even be slightly harmful and decrease the potential for the virus to replicate and spread. As far as we know, though, there's been no magic mutation that has allowed SARS-CoV-2 to become a supervirus. COVID-19 was the perfect epidemic storm because the virus appears to have already been highly transmissible when it entered the human population. And the virus is also quite stealthy in terms of people becoming highly infectious before they even know that they are sick. And sometimes even more stealthy than that because some individuals remain asymptomatic throughout the entire infection, never showing symptoms like coughing or fever, and therefore continuing to go about their daily routines. So the virus was able to spread far and wide even without the help of additional mutations. But thinking longer term, I expect that the virus may evolve to become even more stealthy by becoming even less symptomatic, and hopefully less virulent in the process too. Ultimately, evolution will select for strains that are transmitted more. So from this perspective, it doesn't necessarily benefit the virus to be more virulent unless being more virulent also makes you more transmissible. Making your host very sick by coughing a lot or causing severe pneumonia might actually be detrimental to the long-term fitness of the virus, because people with these severe symptoms will quarantine themselves or be hospitalized. We therefore expect the virus to evolve an intermediate degree of virulence that lets it optimize transmissibility while remaining as stealthy as possible in terms of the symptoms it causes. So hopefully we will develop a vaccine as soon as possible, and surely that is our best bet for getting back to anything like normal. There is, however, the worry that even if we develop a vaccine, the virus may be able to mutate and escape immunity generated by the vaccine. This is what happens with flu and a lot of other viruses, so I think it's reasonable to expect the same will apply for COVID-19. In this case, strains that are able to escape immunity will have a large fitness advantage and quickly rise in frequency. And it's possible people who were already infected or vaccinated will become infected again. This doesn't make developing a vaccine any less important. It just means we may have to continuously update and modify the SARS-CoV-2 vaccine, like we do with the flu vaccine. The same phylogenetic tools that we use to estimate fitness and predict which flu strains will become dominant could then also be applied to SARS-CoV-2, allowing us to stay one step ahead by updating our vaccine to target these newly emerging strains. So back in February, I was at an infectious disease conference with a bunch of different epidemiologists and people who study how viruses spread professionally. And we were taking a look at what was going on in Wuhan and the rest of China. And we were all taken aback by how this was causing such a extreme epidemic in China, and also the extreme responses that the Chinese government was having to take in order to control this virus. But I think the general mindset, even in a room full of infectious disease experts, was, well, let's hope that doesn't happen here, because that would be really bad. And I think that general mindset is something that happens over and over again with the emergence of these new viruses. We know it's happened, and we know it's going to happen again. Several different coronaviruses have already entered the human population and began to spread. What's so special about SARS-CoV-2 is only that it's spread so far and wide. And I hate to say this, but SARS-CoV-2 will likely not be the last virus that enters the human population and causes a major epidemic. So what I would like to really understand is, can we use these experiences of having viruses enter the human population, and can we learn from them to say something about what will likely happen in the future? And oftentimes we can't predict exactly what virus is going to show up and upend our entire world, but we can make educated guesses about if a new virus does enter the human population, what will likely happen.